another biochemistry episode. Today we're looking, as the title suggests, at glycolysis. Let's start with a definition. Glycolysis is a series of chemical reactions in which glucose is converted into two molecules of pyruvate, with the paramount goal to generate ATP. The reason for this is quite simple. All cells require a constant supply of energy to sustain not only their homeostasis, but, it's important to survive. And a huge amount of the energy that is needed is generated by breaking down bonds in a chemical component called glucose. You may know it as sugar. Glucose can be transported into cells via concentration gradients, but way more effective are integral proteins. They're called GLUT proteins. As with many other proteins, there are various different classes. For example, we've got GLUT2, which is responsible for transporting glucose between the liver and the blood, and which functions in an insulin-independent way. There is also GLUT1 and GLUT4, which work in an insulin-dependent way, which just means that they need insulin to work properly. The liver has a special feature, which is Glycogenolysis, which is a process where glucose is produced from glycogen. And this glycogen is stored within the liver. It can be up to 1 kilogram. Skeletal muscles also have the ability to store glycogen, but they are not capable of giving something of their supply away. Back to glycolysis itself. It happens in the cytoplasm and we can find it in prokaryotic as well as eukaryotic organisms, which means that bacteria do it, but we do it as well. It is basically the first step in cellular respiration, so usually it's followed by the citric acid cycle and oxidative phosphorylation. Glycolysis itself involves 10 reactions, and as in every other biochemistry lesson, these reactions are catalyzed by enzymes. As I said before, the goal is to generate ATP, which is the universal energy carrier within our body. We can divide glycolysis into two parts, which makes sense because the first part requires energy, whereas the second generates energy. Let's take a look at the first part and the most important enzymes first. Hexokinase is responsible for catalyzing the first step. This first step is necessary to prevent further interaction between the component and the GLUT transporter. So we need to add a phosphate to the glucose molecule in order to prevent it from going out of the cells again. The isomerase reaction is then required for the cleavage, and one important enzyme is the Phosphofructokinase, which is the rate-limiting enzyme, because it's only active when ADP is high and when ATP is low. As you can see here, we've invested 2 ATPs at the end of the first cycle, but as the rest of glycolysis generates 4 more ATPs, we in total get 2 ATP molecules. Important, and what you might keep in mind is that there is a rate-limiting enzyme called Phosphofructokinase. One essential molecule to keep the whole reaction going is NAD+, which can therefore also be seen as a limiting factor. It's converted into NADH during the reactions, which has to be oxidized back into NAD+. This can be done with oxygen, but also without, which is then called fermentation. So here we've got another important, because the basic difference between fermentation and glycolysis is oxygen. As I said before, glycolysis is usually just the first step of a major energy generating pathway. But for erythrocytes, or also in cancer and stem cells, it is the generator of ATP. This is called the Warburg effect. Important again, keep the Warburg effect in mind. In total, one molecule of glucose is converted into 4 ATPs, 2 NADH and 2 pyruvate molecules. The pyruvate will become important in the citric acid cycle later on.